think there's a significant opportunity in the space to think about, or what I think about is, how much information can you get from one experiment, and that's a really key, I think, benefit of using techniques and technologies like MAM. We can monitor multiple things simultaneously, right? From peptide mapping to critical quality attributes that might be in the form of post-translational modifications. So it's really exciting. I mean, I think, there is some challenges in terms of, it's getting better, but there's still some challenges in terms of understanding what MAM might include. And for example, oftentimes I read and see things that talk about MAM that don't include new peak detection, which I think is a key component of MAM. And then also thinking about, streamlined methodologies that can be tweaked per product, because I realize every product is different, but really thinking about, what are the best parameters for example, for new peak detection, and how do you implement those, especially across the product lifecycle, because I think there's significant uptick or uptake early on, but getting into the quality spaces is also a challenge, I think, for many companies as they grapple with changes and levels of expertise in that space.
Definitely agreed on the new peak detection. That is something that I think is a significant challenge as we're considering MIM. And overall, the main impact for us, we would start looking towards, maybe a reduced release testing panel, so you're not having to test so many different assays per product. So that's a huge, time savings and efficiency gain that would be really a wonderful, progression of this. But a lot of challenges as well, though, is also kind of building that bridge between, how we've been testing versus how we want to test, and showing, comparability, and that it does have the same, quality to the data as what we've been doing previously. So definitely navigating that is a little bit challenging, and it takes a lot of upfront work. So I think really, just the time-wise to put in order to actually show that, MAM is feasible versus how we've done it before.
MAM certainly has potential because you can monitor multiple attributes, and then with agency focusing more on quality by design approaches, that's where having MAM will help in getting a better understanding of the process and product. So right from the beginning, you can monitor, you can characterize all the attributes, get the information, you can quantify, identify, and certainly with the new peak detection, you will be able to really compare and see what's there, which is the outlier. So I think those are some of the benefits, because you can do the faster characterization when you are combining all these techniques together. Talking about the inequality space, yes, it's challenging because mass spec is a sophisticated instrument, so it requires the kind of skill you need to implement there. And then having to monitor under the compliant environment, it's not that easy.
So clearly the use of MAM for product development is a great advantage compared to the combination of other methods, including LC-MS, regular LC-MS, but also all the methods based on chromatography. It helps developing your products more quickly thanks to a single method that will give you information about many attributes, such as glycosylation, deamidation, oxidation, and so on. You can also have access to very high-level information about the sequence. So clearly, in product development, it's a method that can be very, very useful. The challenges to implement that are related to the equipment, the software, that needs to be quite reliable and in order to give reproducible results. But today the main challenge I see is the translation of the use of MAM from product development in process optimization, for example, to the QC. It would be nice to have a method that can be used also in QC, and that's already the main challenge for me today.